So we have now methods to study biopsychology. The preceding section introduced to the nature, like previously we learned about the nature, scope, and major divisions of biopsychology. Now we're gonna study the methods to study. Okay, the structure of the brain is quite complex, and understanding its working is all the more challenging for the researchers. Like for researchers, it is very difficult to study the structure of brain. So with the discovery of Broca's area, it was concluded that different brain areas had different functions. Okay, Broca's area was discovered by Paul Broca. It is a region of the brain responsible for language, actually. Okay, he was a French neurologist, found that one of his patients who had lost the ability to speak had damage in left frontal cortex. More patients with loss of speech show damage in and around these areas, which is now known as Broca's area. So Broca's area is basically responsible for language. Thus, the research conducted in this area concludes that brain damage causes effects like increase or decrease in hunger, changes in emotional responses to to mention just a few. Then we have ablation method, the first method. Ablation means removal of a brain area. Generally with a surgical knife, it is different from lesion. Both ablation and lesion methods are invasive and involve cutting a part of brain. Ablation method thus involves the destruction of some tissues in the brain. This is followed by assessing the changes that occur in behavior due to removal or destruction of the tissue. Any wound or injury thereby is known as a lesion. But one has to be very careful as different parts of the brain are all connected to one another and work in conjunction with another. So, if a lesion is created in one area, it may exert an influence on the neighboring areas as well. Sometimes the animal also recovers from the brain damage either partially or the function of the damaged part of the brain are covered up by another part of the brain. There are different ways to cause lesions. The main technique discussed here are aspiration, radiofrequency, knife cuts, cryogenic blockades, and nerve poison. Okay, then aspiration, a fine thin needle is placed on this tissue, which can be seen by the surgeon and accessible by the instruments. That needs to be examined, and it is aspirated, and the contents are then placed on the slide and examined for any diagnostic analysis. This is generally the preferred techniques when a part of the cortical tissue has to be sucked to reveal the area that is under it for further investigation. So what's the aspiration? In aspiration, we basically a fine thin needle is placed on the tissue which is need to be examined, and the contents are placed on the slide and examined for any diagnostic analysis. Then we have radio frequency. RF current is used to produce subcortical lesions. The wire is placed on the tissue which needs to be examined. The radio frequency devices produce a high frequency current, that is alternating current. The heart that results from this current destroys the tissue in that area. The duration and intensity of the current will determine the size and the shape of the lesion. So RF is basically like it is used to produce subcortical lesions. Like the wire is placed on the tissue which is need to be examined, and the radio frequency devices produce a high frequency current, that is alternating current is used. The heat which comes, that heat will result in the destruction of the tissue in that area. And the next is knife cuts. Knife cuts, like sectioning cutting, is used to make a cut on any area of the nerves of the brain. There is a specific device that is used to make subcortical knife cuts very carefully by placing the brain, causing a stereotaxic apparatus. By using such a technique, extensive damage to the surrounding area is decreased. Like by using knife cuts method, we can prevent the extensive damage. Then we have cryogenic blockade. This is the temporary way of inducing lesions. A cryoprobe is placed in the brain. Through this tube, a coolant is sent to the area under study. The coolant reduces the temperature at its tip to such an extent that the neuron stops sending impulses. Like in this, the coolant is sent to the area, which is need to be studied, and the coolant reduces the temperature at its tip to such an extent that the neuron stops sending impulse. Since the temperature is restored to the normal level, the neurons start functioning again. The temperature is maintained at above freezing point. There is no structural damage. So the thing is also considered that the no damage should be occurred. The next is nerve poison. In this, certain chemicals such as scenic acid or ibotonic acid are used as nerve poisons to create lesions in the particular area in the brain. These chemicals are inserted through a glass tube known as cannula. The destruction is only partial, that is, only the cell bodies are destroyed. So, if there is any change in behavior, then that can be attributed to cell bodies. So, in nerve poison, we use ascanic acid or ibotenic acid, which create lesions in a particular area in the brain. These chemicals are inserted through a glass tube known as cannula. The destruction is only partial, that is, only the cell bodies are destroyed. Yes. So, this was all ablation methods. Okay.